Hello, welcome to Study Hub Pro. Today, we're going to open the case file on one of the greatest mysteries in all of science, the hunt for the blueprint of life itself. For decades, the true culprit was hiding in plain sight, while the entire scientific community was chasing the wrong suspect. So let's follow the clues together.
So, what is it that actually makes a bacteria or a person a person? I mean, what molecule holds that instruction manual that gets passed down from one generation to the next? Now, today, the answer seems super obvious. It's DNA, right? But for a very, very long time, this was one of the most profound puzzles in science.
Our story begins not with the right answer, but with a classic case of mistaken identity. And like any good mystery, the most obvious suspect? Well, it turned out to be the wrong one. For decades, the scientific community was absolutely convinced they had their culprit. And that prime suspect was protein. And honestly, it just seemed to make more sense at the time. Proteins are incredibly complex molecules. They're built from 20 different amino acids, which can create this huge variety of shapes and functions. DNA, on the other hand, is built from only 4 basic units. It just seemed way too simple, kind of repetitive, to hold the rich complexity of life's code. So, the first real break in the case came pretty unexpectedly in 1928 from a British pathologist named Frederick Griffith. The funny thing is, he wasn't even looking for the genetic material, but his experiments with pneumonia-causing bacteria, they were about to uncover a pivotal clue. Griffith's experiment was just so elegant. Okay, so he worked with 2 strains of bacteria, a deadly S strain and a totally harmless R strain. He injects the deadly strain into a mouse. The mouse dies. He injects the harmless one, the mouse is fine. No surprises there. Then, he killed the deadly bacteria with heat and injected it, and the mouse lived. But here's the twist. He mixed the live harmless bacteria with the dead harmless bacteria and injected that mixture. The mouse died. Something from those dead cells had literally transformed the harmless bacteria into killers. So, Griffith had proved that some kind of substance, which he called the transforming principle, could carry genetic traits from one bacterium to another. A critical clue had been found, but the identity of this transforming molecule, it was still a complete mystery. So the case went cold for over a decade. Then, in 1944, a team led by Oswald Avery decided they were going to systematically hunt down Griffith's transforming principle and finally expose its identity once and for all. Their approach was just brilliant in its simplicity. It was a process of elimination. They took the extract from the deadly S strain bacteria, the stuff with the transforming principle in it, and then they treated different samples with enzymes designed to destroy one specific type of molecule at a time. So one enzyme would destroy protein, another would destroy RNA, and a third would destroy DNA. And the results were just stunningly clear. When they destroyed the proteins, transformation still happened. When they destroyed the RNA, it still happened. But, and this is the key, when they used an enzyme to destroy just the DNA, transformation stopped. The harmless bacteria stayed harmless. This was the smoking gun. The transforming principle was DNA. Case closed. Not quite. The scientific world was still pretty skeptical, and a lot of people were still clinging to that protein hypothesis. It was going to take one more definitive experiment in 1952 by Alfred Hershey and Martha Chase to finally settle the debate for good. They worked with something called bacteriophages. These are viruses that are basically tiny little syringes for genetic material. And their trick was super clever. They used radioactive labels. See, they knew that DNA contains phosphorus but not sulfur. So they tagged the viral DNA with radioactive phosphorus. They also knew the virus's protein coat has sulfur but not phosphorus, so they tagged the protein with radioactive sulfur. This let them track each component completely separately. Now, the genius of this whole approach really lies in how these viruses work. A phage is basically a genetic syringe. It latches onto a bacterium, injects its genetic material inside, and leaves its empty protein coat just sitting on the outside. That physical separation of DNA and protein was absolutely key. And here is my absolute favorite part of the story. How did they separate those protein coats from the bacteria after the injection? With a kitchen blender. I'm not kidding. A regular wearing blender provided just enough force to knock the phage shells off the bacteria without bursting the cells open. It's such a fantastic example of using a simple tool to answer a profound question. The verdict was just undeniable. after blending, they found that the radioactive protein was left outside the bacteria, just floating in the liquid. But the radioactive DNA, that was found inside the bacterial cells. And crucially, those bacteria then use that DNA to produce a whole new generation of radioactive viruses. DNA was the genetic material. Case closed. For real this time. So with the case of the genetic material finally solved, a brand new race began. If DNA was the blueprint of life, what did it actually look like? How could this seemingly simple molecule store so much information? The focus now shifted from what it was to how it worked. You know, this final breakthrough didn't just happen in a vacuum. It was really the culmination of nearly a century of work, all the way from Friedrich Miescher first isolating this new substance from bandages in 1869 to Chargaff figuring out these key rules about its composition. Every single discovery was a vital stepping stone. One of the most critical pieces of evidence came from this image right here, photo 51. It was captured by the brilliant scientist Rosalind Franklin using a technique called X-ray diffraction. That really distinct X pattern you see, that was the unmistakable signature of a helix, and it provided the final crucial measurements needed to build an accurate model as DNA. Using Franklin's data, James Watson and Francis Crick were able to piece the final puzzle together. The result, published in 1953, was this elegant and now totally iconic structure, the double helix. The mystery was solved. This shape perfectly explained how DNA could store information and replicate itself, and it opened the door to the entire field of modern genetics. The discovery of the double helix was the end of one great scientific mystery, but really, it was the beginning of countless new questions. I mean, knowing the structure of life's code is one thing, but how does the cell read it, copy it, and, maybe most importantly, protect that precious information from damage and decay? We'll have to save that for another time. See you in the next video.